Today's topic is about beta oxidation of fatty acid, or simply how fatty acid is metabolized inside our body and giving lots of energy or lots of ATP generation. So basically metabolism of fatty acid is called beta oxidation because the oxidation and splitting of two carbon unit occurs at the beta position of the fatty acid. The oxidation of this hydrocarbon chain occurs by sequential cleavage of the two carbon atom, that is called acetyl CoA. So from the fatty acid or acyl CoA, every time acetyl CoA will be separated with the help of beta oxidation. So first we will look over the overview, then in detail of the beta oxidation. For example, for the metabolism of fatty acid, one fatty acid palmitic acid, which is 16 carbon fatty acid, is given. So palmitic acid, that is 16 carbon, so it is first converted into the palmitoyl CoA. So by the attachment of the CoA SH, in detail of CoA SH, that also we will look. So this palmitoyl CoA that will undergo the beta oxidation. So from the beta carbon, 2 carbon fatty acid is separated as the acetyl CoA. Now when it is acetyl CoA is removed, the remaining, that is 14 carbon. So every time beta oxidation will occur and one acetyl CoA molecule will be separated. So ultimately palmitoyl CoA that will provide 8 molecule of acetyl CoA, and this acetyl CoA will undergo the TCA cycle for the further metabolism. But before that, you have to understand about the beta oxidation, and in beta oxidation also you have to understand about the preparatory step. For the preparatory step, initially you have to remember about coenzyme A. So coenzyme A is a complex molecule containing pantothenic acid. It is made up of pentoic acid plus beta alanine and pentoic acid, that is vitamin B5, as well as there is the attachment of beta mercaptoethanolamine, that all together making a coenzyme A. So this SH group over the coenzyme A, that is make thioester bond in the acyl CoA molecule. So acyl, that is simply a word for the fatty acid representation. Acyl means any fatty acid. To emphasize the function of this SH group of coenzyme A, so coenzyme A, that is also written as a CoA SH. Now here that structure of that coenzyme A is given. It is made up of the pantothenic acid plus beta mercaptoethanolamine. So this pantothenic acid is made up of the two part. There is a beta alanine as well as that particular pentoic acid. Now this making pantothenic acid, and over that this particular beta mercaptoethanolamine is attached. In this structure there is a ADP is there plus SH group, which is helping for making a functional group of the coenzyme A. So it is written as a CoA SH. Now in detail of that preparatory step, you have to first remember about the activation of fatty acid. So this activation is occur when fatty acid is attached over the coenzyme A, and for that CoA acyl CoA synthetase, the synthetase word for the utilizing of the ATP. But during this phase, ATP is converted into the AMP, and two high energy bond will be utilized in the presence of magnesium. So acyl CoA synthetase, that is also called activation of fatty acid. So fatty acid is ultimately converted into acyl CoA by attachment with the CoA SH group. Now this acyl CoA, that is finally produced, but it is produced in the cytoplasm. So next preparatory step, and for that carnitine, one more molecule is needed. So fatty acid are activated in the cytoplasm, but the beta oxidation is occurring in the mitochondria. So transport of fatty acid through the mitochondrial membrane is essential, and the long chain fatty acyl CoA cannot pass through the inner mitochondrial membrane easily, and for that one transporter is needed, and that carnitine is work as a transporter, which is involved in the transport of the fatty acid. Carnitine is a one type of beta hydroxy gamma trimethyl ammonium butyrate molecule, that is chemical name, but simply you have to remember carnitine is produced with the help of two amino acid, lysine and methionine, which is produced in the liver and kidney. So in the liver kidney, this carnitine molecule is synthesized with the help of lysine and methionine, which is helping for the transport of long chain fatty acid. So it is also interesting about the long chain fatty acid, that also later on look in this video. So once that acyl CoA which is generated in the cytoplasm will attach over the carnitine and make acyl carnitine. Now then it will be transported inside the mitochondria. So first That next, that is preparatory step 3 with the help of carnitine acyl transferase, which is transferred across the mitochondrial membrane. Here that protein, that is translocase, is needed for the transportation. Now once it is inside the mitochondria, there is a one more enzyme CAT2, that is also carnitine acyl transferase 2, that will transfer acyl carnitine and convert this, separate the carnitine molecule. This carnitine will go back into the cytoplasmic cell, but acyl carnitine is attached with the CoA and finally making a acyl CoA molecule. So that is preparatory step 4, and that is translocase activity. So ultimately, which is activated fatty acid is now present in the mitochondria for the beta oxidation. So transport into the mitochondria depends on the carnitine. So fatty acid CoA, fatty acid is combined with the coenzyme A, and that is making fatty acyl CoA. Now it is combined with the carnitine and making fatty acyl carnitine. Now fatty acyl carnitine, with the help of acyl transferase 1 CAT1, that is transported to the, with the help of translocase protein inside the mitochondria. Now fatty acyl carnitine inside the mitochondria, what happen? Attach with the CoA SH and again make fatty acyl CoA. Now during that time carnitine is separated, and again carnitine will go back into the cytoplasmic site, and for that carnitine acyl transferase 2, that is CAT2, is needed. Here a structure of this carnitine is given, that is simply made up with the help of two amino acid lysine and methionine in liver and kidney, that you have to remember. Now some clinical application or importance of this carnitine that you have to remember: medium chain and short chain fatty acid do not require carnitine for the transportation across the inner mitochondrial membrane. So why you have to remember this carnitine? Because so medium chain and short chain fatty acid are easily oxidized without carnitine, but carnitine deficiency is reported in preterm infants. That's why you have to remember, in whom impaired fatty acid oxidation is noted. So in preterm infant, what happen? If fatty acid diet is given, then this long chain fatty acid will not be metabolized in this preterm infant. So more glucose is utilized in that preterm infant and resulting in a episode of hypoglycemia. So more chances of hypoglycemia in a preterm if carnitine deficiency is there. So that is important clinical application you have to remember. Next, once there is a acyl CoA in the mitochondria, it will undergo the beta oxidation. So here we have seen that fatty acid which is activated and converted into the fatty acyl CoA, that is also considered as step 0, where ATP is converted into the AMP and two high energy bond is utilized. So initially two high energy bond is utilized during this beta oxidation of fatty acid. Now fatty acyl CoA once inside the mitochondria. Then step 1, that is called the FAD link dehydrogenase. So what happened? Here a structure of palmitoyl CoA. Here alpha and beta carbon of the palmitoyl CoA. Now these two hydrogen from this alpha and beta carbon is donated to the FAD, so it is making FADH2 with the help of enzyme dehydrogenase. So FAD is converted into the FADH2. You have to remember this FADH2 because it will provide 1.5 ATP when it will undergo the TCA, it undergo the electron transport chain. So you have to remember first step for the energy production, that is FADH2 generation. So from this palmitoyl CoA, two hydrogen is donated to the FAD and ultimately making FADH2. Now, so when it is donating two hydrogen, what happen? It is converted into the unsaturated fatty acid, that is called enoyl CoA. So that unsaturated bond is generated and making enoyl CoA. Now over this enoyl CoA, there is a hydration process will occur, so that H2O molecule is added. So H2O is basically made up of the H and OH. So H molecule is added over this CH group, another OH group that is added over this unsaturated bond. So this enoyl CoA, now it is converted into the hydroxyl acyl CoA. Because it is containing hydroxyl group, it is called hydroxyl acyl CoA. So hydroxyl acyl CoA is generated with the help of hydratase enzyme by adding of a water molecule. Now next, that is NAD plus dependent dehydrogenase. So whatever this hydroxyacyl CoA which is containing hydroxyl group, over that again from that molecule of CH2 and CHOH, two hydrogen is removed and two hydrogen is donated to the NAD plus and making NADH. Again this NADH which is generated, it is helping for formation of 2.5 ATP in a electron transport chain. But when two hydrogen is removed, again what happen? that 2 hydrogen is removed from the single carbon, so it is now converted into the keto group. So it is now called a keto-acyl CoA. So first what happened, initially 2 hydrogen is removed and making FADH2, again H2O molecule is added, again 2 hydrogen is removed and making NADH, and it is now ultimately made up a keto-acyl CoA. Now this keto-acyl CoA that will undergo the thiolase activity, that thiolase enzyme will work, and here interesting is that one CoA SH molecule is also added. So what happened, when this keto-acyl CoA combine with the thiolase, and then during that time acetyl CoA molecule is separated. So acetyl group is separated and this CoA group that is added over the remaining 14 carbon. So that is the last step for the beta oxidation. Cleavage means 2 carbon is separated in the form of acetyl CoA and remaining, that is 14 carbon fatty acid. So 14 carbon fatty acid that is remain, that will again undergo the same process of the 4th step of the beta oxidation. So what happened next, further cycle will continue, 14 cycle that will convert into 12, 10, 8, 6 and 4. When 4 carbon is undergo the beta oxidation, it will make 2 molecule of acetyl CoA. So that is important thing about fatty acid, beta oxidation of fatty acid. Total 1, 2, 3, 4, 5, 6, 7 and totally 8 fatty acyl CoA will be generated when it is undergo the beta oxidation for the palmitic acid. Palmitic acid containing 16 carbons. So when it is undergo the beta oxidation, particularly 8 molecule of acetyl CoA will be generated. So simply palmitic acid needs 7 cycle of the beta oxidation, but it will generate 8 molecule of acetyl CoA. So it give rise to the 8 molecule of acetyl CoA. Every molecule of acetyl CoA when oxidized in the TCA cycle, so give 10 molecule of ATP. So acetyl CoA, it is also a good molecule for providing a large number of ATP. So in detail of energetic that we have to understand. So FADH2 will produce 1.5, NADH that is generating 2.5 molecule of ATP. So when oxidized in the electron transport chain, so energy yield from palmitic acid may be calculated as, and that you have to remember, 8 acetyl CoA which is generated, each molecule that is making 10 ATP, so 80 ATP. Further 7 cycle of beta oxidation, so 7 into 1.5 that will make 10.5 ATP, 7 NADH that will make 2.5. So total 108 ATP is generated after beta oxidation of palmitic acid. But initially when fatty acid is converted to the acyl CoA, 2 high energy bond is utilized, so it is making 106 ATP. So in the initial activation reaction, the equivalent of 2 high energy bond are utilized. So total 106 ATP will be generated per 1 molecule of palmitic acid. So 1 molecule of palmitic undergo beta oxidation, the net reaction is palmitoyl CoA, 7 FAD, 7 NAD plus and 7 H2O water molecule and 7 CoA SH molecule, that is making 8 molecule of acetyl CoA, 7 NADH and FADH2 plus 7 H plus with the 7 NADH is generated. So that is total summary of the beta oxidation. Now this beta oxidation, and for that simply we needed fatty acid. So fatty acid is under the control of hormone. So availability of free fatty acid, that is the main regulation of the beta oxidation. Now the level of this free fatty acid is controlled by the glucagon insulin ratio. So glucagon increase overall free fatty acid level while insulin will decrease the free fatty acid level. Carnitine acyl transferase 1 is the regulator of entry of fatty acid into the mitochondria, but it is only for the long chain fatty acid, not for the short and medium chain fatty acid. So malonyl CoA, that is inhibitor of this CAT1, carnitine acyl transferase 1 activity, is inhibited by malonyl CoA. Now malonyl CoA, that is also a starting molecule for the de novo synthesis of fatty acid. For the during fatty acid synthesis, malonyl CoA is generated. So during fatty acid synthesis, beta oxidation will not occur. So during de novo synthesis or fatty acid synthesis, beta oxidation will not occur or inhibited. So here simply fatty acid to fatty acyl CoA, and this fatty acyl CoA which is giving rise to the acetyl CoA. But this acetyl CoA is generation is needed when energy is needed in the body. So energy level that is indicated with the help of ATP, FADH2 and NADH. When sufficient NADH and FADH2, that will inhibit beta oxidation, and same way malonyl CoA, that is the synthesis of fatty acid. So during fatty acid synthesis also this beta oxidation is inhibited. So two important inhibitor: high energy level in the form of NADH, FADH2, and malonyl CoA, which is responsible for the fatty acid synthesis that will inhibit beta oxidation.